All right, we'd start out today with a couple of review questions before we jump into our adaptive immunity. So let's look back for just a minute, thinking about those different kinds of cells that we talked about last time in the lecture before. Okay, so patients with too few of a certain kind of white blood cell can be helped by the injection of a medication discovered in the 1990s, granulocyte colony stimulating factor. Okay, that's a hint. That helps to decrease the risk of infection. What kind of cells are these patients lacking, which puts them at risk? Anybody want to hazard a guess here? Basophils, that's a good guess. That's probably the second best answer. Yeah, so you're close. Okay, so the key here, granulocyte colony stimulating factor. So if it's needing to stimulate some granulocytes. So basophils and neutrophils are the granulocytes. And there are the most common granulocyte, though, by far, are the neutrophils. Okay, so that'd be the best answer because there's like way more neutrophils. We said 50 to 70% of the white blood cells in circulation are neutrophils. Basophils maybe make up like 1 to 2%. Okay, so little application question there. One other one, this was, we talked about phagocytosis. I'll give you a minute to read that. Think about what you think the correct answer would be. This one's a little more straightforward. Okay, so always read the question carefully. Multiple choice is tough that way. Which one is false? Okay, so they are all true except answer D. Sorry, they're all false. Wait, which of the following is false? Whoops, see, then I did it wrong. That one is true. The one that's false, sorry, is the last one. Always, when you're, a lot of you, I'm always surprised the exams that get turned in. Some people don't mark at all on their exams. Like a question like this, you want to go through and say, oh yeah, okay, this one's true, cross them out. Okay, because some questions, there's always a few questions on every exam that say which one is false, not which one is true. Okay, so toll-like receptors, we talked about how they are a kind of pathogen recognition receptor that's on the outside of phagocytic cells like macrophages. And they are going to recognize broad-based patterns, so pathogen-associated molecular patterns, not thousands of different antigenic fragments. Okay, that maybe would be for B cells and T cells. Question? So they are on the outside of the phagocyte, and when it does phagocytize the pathogen, they can be expressed on that inner phagosome. Yes, they can be inside as well. Yes. Yep. And we think there's only about 10 different kinds of receptors. So that's, some of them are expressed on the inside that would recognize things on the insides of pathogens, like kinds of, recognize the DNA of pathogens. They can be methylated differently or have like single-stranded DNA or double-stranded RNA. So, yeah, good. Okay, other questions before we move on? So where are we going from here? Chapter 17 is a pretty big chapter. We're going to start with a little bit of an overview, and that's going to include not just what is special about adaptive immunity, but what are antigens. I've used that term maybe a little bit before. We're going to define it better. What makes a good antigen, and what are antibodies? What does antibody structure look like, and how do antibodies function? That's kind of where we're going mostly today. Then we'll get into this major histocompatibility complexes. These are the ways that antigen presenting cells, like macrophages and dendritic cells, will present antigen to B cells and T cells. Had the very good question last time, why don't antigen presenting cells get eaten themselves or killed themselves when they're presenting that antigen? Then we'll get to it now, because they're presenting typically to a type of cell that's not acting to kill the cells, it's acting to help other cells. So wait for it, we'll get to that. And then more specifically, how B cells get activated, kind of save the most complicated thing for last, as well as how T cells get activated and what they actually do when they're activated. So that will all lead up to a discussion of vaccines, which I have greatly expanded that part of my talk over the last couple of years, because that's kind of what's in the news right now. So all leading up to that. All right, so what's special about adaptive immunity versus innate immunity? For the most part, our innate responses, like complements and the action of neutrophils degranulating and macrophages phagocytizing, those are going to happen pretty much the same every time those proteins or cells come in contact with a particular pathogen. There's a little, that's not always exactly the case, but that's kind of the traditional view, that those kind of responses are very similar every time it's encountered. When our body has the lymphocytes, the T cells, and the B cells that are able to adapt and act better and more quickly and more efficiently the second time an antigen is encountered. And they also are going to respond to very distinct invaders. So we could expand on this a little bit more, but two especially important attributes of adaptive immunity: specificity and memory. Okay, by specificity, just kind of what I said before, our immune cells respond to one particular or one specific shape. So an important distinction is that we are not crafting a response based on that pathogen. We already have B cells and T cells with receptors that will match any pathogen, any invader that we could possibly come in contact with. So we'll talk about how that diversity is generated. For example, you probably have B cells and T cells in your body that would recognize stingray venom. Are you ever going to come in contact with that? Probably not. But you have the ability to do that, maybe just a few cells, and then those few cells get induced and cloned and made much more of them. Okay, so very specific shapes. And then those cells can make memory cells. So the next time that pathogen is encountered, we have a stronger or a stronger and faster response. Stronger and faster response is going to be generated. Okay, so to illustrate that, we're going to talk about this again later. But say here's your initial response to some pathogen. Over time, what kind of concentration of antibody is generated from that? It usually takes about 10 days or so to really get very much antibody produced. That's one other attribute of our adaptive immunity. It takes time because the right cells have to come into contact with each other to be induced to happen. So it does take some time after initial exposure to a pathogen, but then at some distant time later, if that individual is exposed again to the same pathogen, we get a lot quicker response, a lot more antibody made, and it's probably the right kind of antibody that's best suited to fight that pathogen. Okay, so this is the basis of vaccines. We expose somebody to, say, give them a tetanus shot at some time, and then what the idea is later on in the future, if that individual is exposed to it again, they have such a fast response that they don't even know that they've been exposed to it. I was thinking this week that it would be nice if we got like a notification on our phone that said, hey, good thing you got this vaccination. It's working. You just fought off measles or COVID or whatever. There would be, it just works and we don't even know it most of the time. Same thing, when I was a kid, I got chickenpox. That was my first exposure. That usually infers lifelong immunity. So all the many times that I've been exposed to that later on in my life, I didn't even know it because I had memory cells that responded right away. Okay, so we talked about this a little bit, these cells a little bit last time. It's kind of important, so it's bears repeating. Of course, the lymphocytes, the B cells and T cells. Just so again, a little overview, the B cells are going to mature into plasma cells. And plasma cells are the ones that are kicking out lots and lots of antibody. I think that term sometimes throws people. Of course, plasma is the fluid portion in our blood. That doesn't have really anything to do with plasma cells. Okay, plasma cells are B cells that have differentiated, that have been activated. So I don't know, maybe that term is a little bit confusing. And we said that's called humoral immunity when we're talking about the action of antibodies. Okay, it works in our body's humors, our mucous membranes, our bloodstream. And then T cells, we have a couple different kinds of T cells that work. T helper cells, abbreviate those TH from now on out. They are famous for basically kind of orchestrating the whole immune response. We'll say orchestrating the adaptive immune response. Sometimes an analogy that's used is that T helper cells are kind of the general in charge of the army. Okay, they mediate a lot of different reactions. They activate B cells, they activate other kinds of T cells. So that's the type of cell that is most damaged in HIV infections. So when a person does not have very many T helper cells, they have a very poor immune response to lots of different kinds of pathogens. And then a second type of T cell, cytotoxic T cells, which we'll abbreviate TC. Okay, they are going to target and destroy host cells that are infected with some kind of intracellular pathogen. So there are bacteria that are intracellular that have to be inside of a cell in order to reproduce. But the first thing that we think of usually is viruses. Okay, they start out outside our body, but then they gain access to our cells, and it's very difficult for antibodies. They're not going to be able to notice viruses because they're inside our cells. So we have to have a whole mechanism by which cells are going to advertise that they've been activated, but that they've been infected, so that cytotoxic T cells can act on those cells. And they can't just kill the virus, they have to sacrifice the cell to get rid of it. So much more about that. And again, that's considered cell-mediated immunity. I guess because it's mediated by T cells. Okay. All right. So what do we call the things that our immune system responds to? That's the term antigen, literally antibody generators. Okay, so I've had people say to me before, I can't remember which is the good thing, the antibody or the antigen. Okay, we make antibodies, we generate antibodies in response to antigens. So let's see. So let's say they act to activate immune defenses. So what kinds of things act as antigens? Pretty much any kind of a pathogen, parts of viruses, fungi, protozoa. Other things can act as antigens that are not microbes. So things like dust or the feces of dust mites can act as an antigen and elicit an inappropriate immune response, right? Allergies, food, shellfish, peanut butter can act as antigens and cause, again, kind of an immune response that we don't want, unfortunately. Now, often we're talking about one antigen. So say, for instance, Staphylococcus bacterial cell actually has many different epitopes on it. So it has many different shapes that our immune system can react. So epitopes, let's define that as regions of antigens that are recognized by antibodies. Okay, so the picture here, sometimes I kind of use those terms a little bit interchangeably, but say here the bacterial cell is acting as an antigen. There may be many different proteins on its surface that many different antibodies or B cell receptors or T cell receptors will recognize. Okay, so something that's really antigenic, meaning it's eliciting a really strong immune response, is probably going to have lots of different epitopes, lots of different things that our immune cells and antibodies are going to recognize. Okay, so what makes something a good antigen? And again, what I mean, if it's a strong antigen, that elicits a real strong immune response. Couple different things. We look at the shape, or kind of related to that is the complexity of the molecule. If the molecule is really has very complex 3D shape, then it's going to be more likely to be recognized. The more complicated, the more epitopes there are. The other thing we will look at is the size of the molecule. In general, really large molecules are more likely to be antigenic. A very small molecule is typically not going to be antigenic, but there are exceptions to that, because sometimes really small molecules will actually link up here with some carrier protein, for example, and then can be noticed by our immune system. So those really small molecules that do that, we refer to them as haptens. So something like poison ivy oil is actually such a small molecule that it shouldn't be antigenic. But if you've ever had poison ivy, you know that it is. That oil, when you brush against a poison ivy leaf, when it soaks into your skin, urushiol is the name of the compound, I think. It actually complexes with a carrier protein when it soaks into our skin, and it gets noticed that way. Penicillin, metabolized penicillin, also is a really small molecule, shouldn't be antigenic, but it can complex with carrier proteins, again, when we metabolize it in the liver. So for some people, that acts as a hapten as well. Okay, so what kind of molecules are going to be antigenic? Proteins are generally the best antigens because they're large molecules and they're very complicated. So here's an example of an enzyme. It's the enzyme beta-galactosidase, the one that splits lactose and its two monosaccharides. So it just, note the complexity, lots of different epitopes there. Proteins are typically going to stimulate both humoral immunity and cell-mediated. Okay, going to stimulate both B cell activation as well as T cell activation. Carbohydrates, much less antigenic because here's a typical carbohydrate starch. It's just many glucoses, all in a line. So it's the same, one structure repeated over and over again, not as complicated. So not as many epitopes. Carbohydrates, like the capsules of bacteria, can be antigenic. Typically, they're going to stimulate a type of humoral immunity only. And we'll get to examples of that in a little bit. Other things like lipids and nucleic acids are usually not antigenic unless they're complexed with a protein or a carbohydrate. So something like a glycolipid or a nucleoprotein could have some antigenicity. Okay, so, all right, so a little more terminology. We're almost done talking about antigens, but a little bit more terminology that we use. We're talking about kinds of antigens. And I think I used this term last time when I was talking about fever. Anything that's an exogenous antigen, that just means it comes from outside of the body. Okay, so I think I used the term an exogenous pyrogen would be a part of a bacterial cell, like lipopolysaccharide, for example, that acted on the hypothalamus to change our body temperature. So an exogenous antigen, that could be a virus that you breathe in. It could be a bacterial cell that infects a wound. And then an endogenous antigen is one that's produced within the cell. So viruses are going to start out exogenous. Once they gain entry to our cells, then they're going to be endogenous. Okay, so the way our immune system detects those antigens, it depends. Certain cells are better at detecting the exogenous ones. We have other mechanisms to detect ones that are inside of our cell. An autoantigen is usually a protein that's part of our self. Okay, so under normal conditions, our self proteins or our self molecules should not elicit an immune reaction. So let me note that. So under normally, do not elicit immune reaction. But in some cases, our normal immune tolerance has been lost. That's part of B cell and T cell maturation, that they lose receptors, that are going to be specific for autoantigens, because we don't want a reaction to ourselves. But sometimes that maturation doesn't happen correctly. There's autoimmune disease, that tolerance is lost, and sometimes our immune system targets our own cells. And then we have autoimmune disease. So here's an example of one of many autoimmune disease, rheumatoid arthritis, where we've got antibodies that are targeting the joints, for instance, can be very debilitating. And then one other kind of antigen that we're going to kind of circle back to at the end of our discussion, maybe next time, are superantigens. And usually we would have, like I said, very specific B cell or T cell receptor that would only recognize one very specific thing. But in some cases, there are some, say, like the toxin from toxic shock syndrome that's produced by Staph aureus, that acts as a superantigen. And that means that there are many, many different T cells that would recognize it. And you think, well, that might be a good thing. Then we have a quick response to it. But in fact, it's bad, because when so many T cells recognize the same thing and they're all producing cytokines, it causes just an overwhelming inflammation. So, too much inflammation, way more than what's appropriate, and that can cause the patient to go into shock, which I kind of forgot to talk about that when we talked about inflammation. But if our blood vessels are vasodilating for too long and too many cells, too much fluid is leaving the bloodstream, the blood pressure can go way down. Okay, and that's when the patient goes into shock. They're not getting enough oxygen to their organisms, and multiple organs can start shutting down. So that's a bad thing. So superantigens, really bad. In those cases, patients have to be given anti-inflammatories in addition to trying to get rid of the pathogen. Okay, so antibodies are what are generated from an activated B cell. And we'll talk more about how that activation takes place a little bit later. But I want to first talk about the different kinds of antibodies, their basic structure, and what antibodies actually do. So antibodies are a kind of immunoglobulin. And in fact, most of the time we use those two terms kind of to mean the same thing. So immunoglobulins, those are antibodies, those same thing. They are glycoproteins. They're mostly protein. There are a few carbohydrates that are kind of associated with the stem portion of the antibody there, and that's why they're glycoproteins. But mostly peptides, and they're composed of 2 heavy chains and 2 light chains. So here's the heavy chains here. That just means they have a higher molecular weight. And then here's the 2 light chains outside of that. This region, which sometimes we refer to as the ARM region, is also called the FAB, or the FAB region. That stands for fragments of antibody binding. Okay, so this is the business end of the antibody. Right here and here, that's where the antibody is binding to epitopes of antigen. Okay, so we'll talk about, well, what happens when it binds there pretty soon, and the help that does. But this is the variable region of the antibody, and that shows up a little bit better when we see a space filling model here, where basically every atom is a little ball in this model here. So that kind of helps us understand the FAB region there. It's kind of like a jigsaw puzzle. It has to fit in just right to recognize the antigen. So many, many, many different possibilities, slightly different shape, and then the antibody's not gonna bind. Okay, so tremendous variability at the end of the FAB region. And I should say both sides, they're going to recognize the same antigen epitopes. And it seems like, wouldn't it be more efficient if each antibody arm could recognize a different thing? Perhaps, but one of the functions of antibodies is to clump antigen together. So that's going to happen better if it's recognizing the same thing and clumping a bunch of the same antigen together. All right. There's a hinge region. So basically, if you're thinking about your arms in the air, you bend at the waist. That hinge region is a little bit flexible. So I don't know, it can kind of turn around and bind to different things. And then the stem region here, we refer to that as the FC portion. That stands for fragment that's crystallizable. It might not be all that helpful, but there are basically 5 different regions on the gene that gets transcribed and translated into antibodies. So those gene regions are named for Greek letters like mu, gamma, alpha, epsilon, delta. Whichever one of those stem regions gets translated into the protein, that makes up the 5 different classes or isotypes of antibody. So IgM, IgG, IgA. Okay. All right. So that's a basic structure of the antibody. What are the different classes of antibody? They're all they kind of have their things that they're good at, the different classes, the isotypes. What IgM is famous for, this is always going to be the first antibody produced. Okay, so once those B cells differentiate into plasma cells, those plasma cells are always kicking out IgM antibody at the beginning of an infection. It's only about 5 to 10% of the total antibody in the blood. That's because they're fairly short-lived. They don't stick around for very long, and they're typically going to be replaced by a kind of antibody that may be better suited to fight against whatever infection it is. So they're quickly replaced. They are a pentamer in circulation. So basically 5 IgM antibodies come together and they're joined by this polypeptide called a J chain. So this is the J chain right here. That has some role in joining those 5 individual IgMs together. And so basically the IgM is going to have 10 different binding sites. Different might be the wrong thing to use, but remember, it's all the same specificity. In other words, each arm there is recognizing the same antigen. So again, it can be really good at clumping lots of antigens together. Oftentimes, plasma cells then will soon after it starts making antibodies, will get cytokine signals to change the isotype or the class of antibody that's made. So again, soon after it starts, that it'll get the signals to class switch and to a different kind of antibody. Most often, class switching is going to happen in terms of switching to the IgM antibodies, because IgG. My brain was thinking one thing and came out of my mouth is different. Because 80% of antibodies in circulation are IgG. So this is the most common antibody in circulation in the bloodstream. So that's what I mean by circulation. And it's the most common because they typically stick around for a while. Okay, so they're the longest acting. So it makes sense. Sometimes you got to memorize stuff, but sometimes you got to think about, well, what makes sense? It makes sense that this is the kind of antibody that's going to cross the placenta. Okay, this is the one isotype that's able to do that. And that makes sense because during development, the fetus can be basically any pathogen that the mother is exposed to, she's going to make antibodies and share those with her baby. So those antibodies will persist during the first few months of life after the baby's born. Okay, so babies that are born prematurely don't have as much access to mother's antibodies. But usually that's going to be a good thing, right? That the baby, that the mother shares some antibodies with the baby. The one time that that's detrimental is when there's problems with Rh disease. Okay, so everybody has, you know, with their blood type, you either have that rhesus antigen or you don't. So for instance, I'm A positive, so I'm positive for that rhesus antigen. So when I had my kids, that wasn't any problem. I did not make antibodies to that rhesus antigen because that was something that I had already. Now, other people, say my daughter, who's O negative, she does not have the rhesus antigen. So if she was pregnant and her baby did have the rhesus antigen because, say, the father was A positive or O positive or whatever, then she may start making antibodies to the baby's blood cells. All right, so that's usually not a problem in a first pregnancy, but if there's usually a little mixing of maternal and fetal blood during birth of the first one, and then subsequent pregnancies, the baby's much more likely to be targeted by the mother's antibodies. Luckily, we have RhoGAM shots that can be given to the mother at certain times during the pregnancy, so that doesn't cause, you know, fetal anemia or hemolytic disease of newborn, which can be fatal. So that's just the one instance where the mom's antibodies are not good. Okay. Sometimes we get class switching to IgA antibodies. Okay. And these are often present as a dimer, especially when they are secreted onto mucous membranes. Okay, so this is going to be the main antibody present in mucous membranes. Okay. Although, so it's only about 13% of antibody in the bloodstream. Okay, but it's the dominant antibody that gets secreted onto mucous membranes. There is this secretory component that allows the antibody to leave the bloodstream and enter those mucous membranes. It also, that secretory component does afford some protection to the protein against proteolytic enzymes that might be on those secreted surfaces. And once again, there's this J chain that forms a dimer that makes the two antibodies kind of attached to each other. Okay, so because it's the main antibody on mucous membranes, it's also going to dominate in some bodily fluids, things like tears, saliva, as well as breast milk. Okay, so that secretory component is really important. Those antibodies in breast milk, when they are drunk by the baby, right, the milk goes through the baby's stomach, okay, very acidic, lots of proteolytic enzymes in the digestive tract. So that secretory component keeps the antibody intact so that it can do some good for the newborn baby. All right, the last two make up a really small percentage of total antibody in circulation. IgE, we know that it can bind to receptors on basophils and eosinophils, granulocytes, and cause them to degranulate. So this most often happens when the body is fighting some helminth worm infection. So we talked about how eosinophils, how they degranulate on parasitic worms. Again, they have receptors for IgE. And we also find a little higher percentage of IgE in the bloodstream when there's some kind of allergic reaction. Okay, so kind of, yeah, you know, these days you think, ah, what do we need IgE for? I don't know, maybe not as much. And then IgD, we don't know a lot about IgD. It doesn't appear to ever actually be secreted. So IgD antibodies are basically just, they stay attached to B cells and act as B cell receptors. It doesn't appear that all animals have IgD, so we're not sure really their importance. There are animals that have other types of antibodies. Sharks have IgW, for example. So there is some variation there. Okay, what do antibodies actually do? We have 5 different functions of antibodies. One big one is neutralization. So antibody acts to bind to adhesion molecules of pathogens. We'll say, or toxins. Okay, and then when it binds to those adhesion molecules, then the pathogen can't adhere to its target cells. So here's, for example, some antibody binding to some spike proteins of a virus. Maybe that's COVID, binding to those spike proteins. Now the virus has a much harder time attaching to its target cells. We could have antibody that binds to a toxin. Again, and then the toxin can't act, so coats the outside of a toxin. Here, this looks like a bacteria flagella. So a flagella is not an adhesion structure. Maybe if it was fimbriae, this would make more sense. I suppose if antibody, coated the flagella, then it would keep the bacteria from moving, maybe where it needed to go, and that could do some good as well. Okay, so that's a big one. We know that some antibody can act to activate complement, and then indirectly, which again, complement causes inflammation as well. So that was one of the 3 different activation pathways of complement, how antibody and binding to an antigen antibody complex. Sorry. So yeah, so antibody binding to, a pathogen with antigen on the surface will act to make C1, then act to split C2 and C4. Okay, so that's going to, again, let's see, activate that classical activation. And then, of course, the opsonization and the inflammation, then there'd be less of that. So we could say, either opsonization directly or indirectly by activating complement, or but so remember, opsonins are things that are going to coat the outside of pathogens to make it more likely that they will be phagocytized. So we see how that works. Here's a pathogen. There are FC receptors in the outside of the phagocyte. Say this is a macrophage. Remember, FC, that's the stem region of the antibody. So if the antibody's coating the outside of the pathogen, then now the macrophage can just get a handle on that pathogen, and it's going to be much more likely going to stimulate phagocytosis. Okay, even if that pathogen is coated in a capsule or something like that. Okay, helps the pathogen adhere. A 4th function I alluded to already, agglutination. Okay, that makes the antigen stick together. Okay, so the immune system is much more likely to notice a big clump of antigen, and that can get filtered out either at the lymph nodes. We'll talk more about different structures of our anatomy that have immune function pretty soon, lymph nodes, or it can get filtered out at the spleen. Okay, so the spleen filters blood, lymph nodes, lymph is going to filter through there. Okay, again, get a big clump, that's going to get noticed. And the last function of antibodies that we'll talk about involves the help of natural killer cells, NK cells. Okay, so here we have some pathogen being coated in antibody. Natural killer cells have, again, Fc receptors, receptors for the stem of that antibody. This antibody-dependent cellular cytotoxicity is most likely going to be a factor if there's antibody coating some especially large pathogen, one that couldn't, too large to be phagocytized. So the NK cell, when it binds to these antibodies, that stimulates the natural killer cell to release cytokines that are going to act to cause the death of the pathogen. Okay, and it's very similar in the way that, as we'll see, that cytotoxic T cells actually act on our host cells that are infected. Questions so far? Just kind of been throwing a lot of information at you. This is a table that kind of sums up the different isotypes. Notes, hey, here's IgG. That's the most common antibody, and that's the only one that crosses the placenta. In terms of what the antibodies actually do and which isotypes are best at certain functions, I would say don't worry about that so much. I wouldn't expect you to kind of know or memorize all of that, but it just points out that, hey, some isotypes are better at certain functions, some isotypes are better at other functions. Okay, so let me just start to introduce here today the MHC glycoproteins. This is getting into a little bit more complexity, but it's important to understand this before we can talk about how B cells and T cells get activated. Okay, so this is the mechanism that our cells have to kind of advertise that they are infected with some kind of pathogen. Okay, they're called major histocompatibility proteins. They're surface glycoproteins that span the cell membrane, and they're coded for by a cluster of genes called the major histocompatibility complex. And I bring that up because this gene complex is considered polymorphic. And that just means that there's a ton of variation between individuals as to what kind of MHC glycoproteins they produce, because we all have a little different variation of that gene. And that's important to understand because when two people are infected with the same pathogen and they're both healthy and the same age, and one person gets sick and one person doesn't, it's because they have different genetic responses. So one way that they're different is one person may present just slightly different epitopes than another person. So one person could actually have a stronger immune response than the other. So genetic differences, this is one place where that comes up. In humans, we call these genes human leukocyte antigens. Or that's often abbreviated HLA. Okay, so sometimes sometimes you might be reading a paper and it's talking about HLA glycoproteins and, oh, okay, they're talking about MHC. Just call it something different in humans. And like I said, their main function here is to hold parts of antigens for presentation to T cells. Because it turns out that T cells can't recognize antigen by themselves. They need it presented in a very particular way. It has to be presented to them by these glycoproteins. Okay, so there's two different kinds, and they have a very different function. MHC class 1 is present on all body cells that are nucleated. So what kind of cells in our body don't have nucleuses? One specific kind that I'm thinking of. Okay, red blood cells. Okay, so red blood cells had a nuclei, and at some point during their development, they lose them. Apparently, they don't need them. Okay, so MHC class 1, all of our body cells present antigen with MHC class 1, except for red blood cells. Okay, and they are going to present antigen that's, again, inside our cells to cytotoxic T cells. Okay, we're kind of in the middle of something, but we're also out of time. So I'm going to kind of go through this again next time, and we'll pick up from there. I hope you all have a good weekend. Okay, and don't stress over your nursing stuff.